Hello, my name is Bhanwar Lal Puniya and I am going to present my research about metabolic drug repurposing for autoimmune diseases. I'll be focusing on using the metabolism of CD4 plus T cells as therapeutic targets for three autoimmune diseases. Those are rheumatoid arthritis, multiple sclerosis, and primary biliary cholangitis. So CD4 T helper cells are white blood cells that are essential part of the human immune system. CD4 T cells receive signal from antigen presenting cells and become activated, and then they pass this signal to cytotoxic T cells and natural killer cells, which will secrete perforins and gray enzymes in cell-mediated immune response to kill infected cells. CD4 T cells also pass signal to B cells that will make then plasma cells, which will secrete antibodies, and these antibodies will bind to antigen to neutralize them in humoral immune response. So CD4 T cells are highly versatile, they are polyfunctional, and they can differentiate into one of the several functional subtypes in response to context-dependent signals. There are four classical subtypes of CD4 T cells, T helper 1, T helper 2, T helper 17, and regulatory T cells. Th1, 2, and 17 are effector T cells, and they are responsible for generating immune response against different infections and diseases. On the other hand, regulatory T cells, they are responsible for suppressing clonal expansion of effector T cells. Therefore, they have immunosuppressive functions. So during the immune response, when CD4 T cells are activated, they will proliferate. And during the rapid proliferation, there will be increase in bioenergetic and biosynthetic demand. And there will be accumulation of cell biomass. And to fulfill these demands, there will be metabolic rewiring and adjustment in the metabolism of T cells. So if we compare the metabolism of effector T cells with the naive resting T cells, effector T cells are highly glycolytic and they also use glutaminolysis. On the other hand, naive resting T cells, they use oxidative phosphorylation and fatty acid oxidation for energy generation. So CD4 T cells help protect against infections and diseases. They are also associated with many diseases, including autoimmune disease and cancer, if dysregulated. In these diseases, the metabolism of CD4 T cells was also found as dysregulated. In multiple sclerosis and systemic lupus erythematosus, more than one type of effector T cells are found as dysregulated. Therefore, we aim to explore CD4 T cells metabolism as a target for autoimmune diseases. And for that, we used a genome scale metabolic modeling. And in the first step, we analyzed transcriptomics and proteomics data. We performed multiomics data integration and identified different active genes across four types of CD4 T cells. And we use Recon 3D as reference metabolism. So Recon 3D is a generic metabolic model for human, and we used with the data we analyzed, and we constructed context-specific metabolic models for four different T cell subtypes. And in these T cell submodels, there were 5,000 reactions and around 1,000 genes in each model. I'm showing an example of flux distribution in central carbon metabolism. This map shows that T cells take up glucose, and then glucose will be converted to lactate in the aerobic glycolysis pathway, and this is also taking up glutamine here, that is converting to alpha ketoglutarate in glutaminolysis pathway. These are characteristics of activated CD4 T cells which are shown by the model we constructed. We have also validated the model by reproducing specific behaviors of CD4 T cells. For example, all the models, they showed some dependencies on glucose. When we reduced the uptake of glucose, there was reduction in the growth rate and biomass production, and that was observed in all 4 models. In the second example, we increase flux through pyruvate dehydrogenase, and we observed the reduced production of lactate that was also in agreement with the experimental observation. Overall, our model reproduced 8 specific behaviors and predicted many genes as critical for T cell growth and proliferation, which was in agreement with experimental data in the literature. So we wanted to use these metabolic models to identify drug target, and for that first we analyzed transcriptomics data sets from case control studies, here patients and healthy from 3 autoimmune diseases, and we identify differentially expressed metabolic genes and group them into downregulated and upregulated genes. In the model, we mapped drug targets and compounds from drug databases, and then we introduced the perturbation. Under each perturbation, we identified reactions with increased flux and decreased flux, and then we compared the downregulated genes, and we find that how many of downregulated genes were become up when we introduce this perturbation, and how many of upregulated genes become down under the perturbation. So by accounting for all these changes and feeding them into scoring function, we came up with a perturbation effect score, and then we compared this score across different T cell subtypes, and finally utilizing this score we came up with the rank of different genes and predicted these genes as drug targets for 3 autoimmune diseases. So we predicted a total of 68 drug targets, and 17 of them for rheumatoid arthritis, 27 of them for multiple sclerosis, and 24 for primary biliary cholangitis. As an example, I'm showing here 2 drug targets, which is catechol O methyltransferase and alcohol dehydrogenase 5 in tyrosine metabolism. The COMT is interesting. It was identified for 2 diseases, rheumatoid arthritis and primary biliary cholangitis, and COMT is associated with multiple reactions in tyrosine metabolism. Next, we experimentally validated some targets. For example, I'm showing results of entacapone. That drug is used to treat Parkinson's disease combined with other medications. So we used entacapone, and that target COMT, and entacapone at different concentration of the drug, we looked at T cell proliferation, and we found that at 100 and 1000 micromolar concentration, the proliferation of T cell was significantly reduced, and that was in 2 different time points, 48 hours and 72 hours after drug treatment. Overall, by combining in vitro experiments and existing knowledge, 50% of the target we predicted, they have been found to suppress CD4 T cell proliferation. And we tested 10 different drugs, and 4 of them has found to reduce the CD4 T cell proliferation. And these 4 drugs are entacapone, icosapentaenoic acid, perhexylene, and fluorouracil. Finally, we came up with this automated pipeline that analyzes transcriptomics data and proteomics data, combine them to identify active genes, and with the help of Recon 3D, we can generate genome scale metabolic networks for specific cell type, and then utilizing different drug databases, we can map these and we can you do the perturbation analysis, and then using the disease data, which is from the case control studies, we can identify differentially expressed genes and group them in up and down regulated, and then compare the results of perturbation analysis with the differentially expressed genes. We can utilize the formula we used in our study to rank different drug targets and to identify repurposable drugs. In the end, I would like to thank my supervisor, Dr. Heliker, and our collaborator, Dr. Barberis, for their guidance, support, and supervision in this study, and Dr. Rada Amin for experimental validation, and Bailey, Robert, and rest of the team to help me with data analysis and developing different models. I would also like to thank NIH for grant to Dr. Helikar and Systems Biology grant of the University of Surrey to Dr. Barberis. Thank you.